So, our objective at this point is to try to assess the carbon budget of a cell and then to try to figure out what that implies about the transport of sugars across the cell membrane. So, the first question that I want to address is how many carbons to make a bacterium. And let me say, this is only the building materials. So, we're not thinking about paying the workers. What I mean by that is, there's carbon skeletons, and that's the part that we're assessing. But in addition to that, the sugars are used to produce energy, which then is used to pay the workers, so to speak. So, so what we want to know is how many carbons to make a bacterium, and the strategy that I'm going to take goes as follows. So I take that the volume of the bacterium is of order 1 micron cubed, or 1 femtoliter, which is 10 to the minus 15 liters. This means the mass of a bacterium is on the order of rho times V, which is equal to 1 kilogram per liter times 10 to the minus 15 liters. So that's 10 to the minus 15 kilograms, or 10 to the minus 12 grams. So a picogram is the mass of a bacterium. Now, the dry mass is something like 1/3 of the mass of the bacterium. So we're going to call that 0.3 times 10 to the minus 12 grams. And my last point before actually trying to figure out how many carbons is to note that the dry mass, I'm going to say roughly 1/2 of the dry mass is carbon. And if you want to think about that, then I could say that how can we see that? So one idea would be to note that a typical amino acid, for my purposes, is about 100 Daltons. And there's usually, let's say, 5 carbon atoms, which is around 60 Daltons. And so, that's my indication that roughly half the mass is carbon. Okay, that's a very slick, sort of sloppy way to go after it, but that tells the principal parts of the story. So, so what do I learn from that? I learned that 15 over 100 times 10 to the minus 12 grams of carbon in a cell, in a bacterium. And the 15/100ths is my way of reporting the fraction that is carbon. And then what I want to do is I want to divide by 12 grams per mole. And so, for my purposes, I'm going to call this basically, what do I want to say? Something like, so I'll just take the 15 and the 12. What do I want to do with those? So maybe we'll call that 5/4 times 10 to the minus 14 moles times 6 times 10 to the 23 carbons per mole, which yields something like, what does that tell me? Something like 5 times 6 is 30 divided by 4. I'm going to call that 7 times 10 to the 9th carbons. And if there are 6 carbons per sugar, this implies something like 10 to the 9th sugars. Okay, so again, I'm doing street fighting. I'm not trying to put too fine a point on these things, but it's roughly 10 to the 9th sugars. Now the next question that I can ask is, how many transporters do I need? So that's the next question. And the way that I would think about that if I were on the desert island with Wilson is I would try to figure out, well, what's the typical rate constant associated with a kinetic process mediated by single molecules? And I would say, the number I would carry around in my head is something like 100 cycles per second, so 100 transported per second. So I'm going to make the claim, I'm just going to tell you 200 sugars per transporter per second. And so the number of transporters needed is going to be something like 10 to the 9th sugars divided by, let's say, 5 times 10 to the 3 seconds. And then, so this is the cell cycle time, the 5 times 10 to the 3rd seconds. And then divided by 200 sugars per transporter per second. What's that going to lead to? That's going to lead us to, so 10 to the 9th divided by 5 times 10 to the 3 is going to give us an 0.2 times 10 to the 6th. And then that's divided by 2 times 10 to the 2. And so, and that's transporters. So I can rethink that as 2 times 10 to the 7 divided by 2 times 10 to the 2 for as many as 10 to the 5th. Oh, wait. What's this, 10 to the 9th sugars? Yeah. So that gives me 10 to the 9th transporters. Something seems a little off here. I would have thought down by a factor of 10. So what did I do wrong? Maybe I made an arithmetic mistake. 0.2 times 10 to the, yeah, sorry. 0.2 times 10 to the 6th is 2 times 10 to the 5th. 0.2 times 10 to the 6th is 2 times 10 to the minus 1. Yeah, that's 2 times 10 to the 5th. And so that gives me on the order of 10 to the 3 transporters. I apologize for my mistake. So, so we conclude that there should be on the order of 1,000 transporters. So this was for a 5,000 second division time. And so if we had 1,000 second division time, we'd need a factor of 5 more transporters, so that would be 5,000 transporters. So that's interesting. That we'll come to the data in a moment. And so what I would say is what we've done here is we've tried to reason out the demands for carbon in the dividing bacterium. And now let me try to examine for you the data that comes from proteomics. So you'll recall that we made the argument that proteomics makes it possible for us to understand what the state of transporters is. And so here at the top, whoa, I don't know what I just did, but that was bad news. Sorry, still working out the glitches here. So I was going to try and circle something with my pen, but that doesn't seem to have worked. So the estimate on the top is basically what I just did. So 0.15 picograms per cell, and we concluded that we got something like 7 times 10 to the ninth carbons. So here I'm saying 10 to the 10th, 5 carbons per sugar, as shown here, and here's the transport rate. And that leads to a prediction of on the order of 2,000 transporters, as you can see here. And what I'm showing you at the bottom is the state of carbon, sugar, glucose transporters. Okay, so that's what's plotted here. And what you can see, the brown dot right here, is the estimate that we just did. And this stuff is the data from the various mass spec and ribosomal profiling points of view. So there's an interesting observation here that I think still needs to be considered in more detail, and I don't have anything particularly deep to say about it. Other than that, cells are poised to do a lot more carbon, sugar, glucose transport than is demanded by the 5,000 second growth rate. Now, let's look at the other parts of the figure, because I think that that's very interesting. For example, this top figure is plotting for us the number of glycerol transporters as a function of growth rate. And the thing is that at the particular case where glycerol is indeed the carbon source, the number of glycerol transporters goes up by a factor of 100, let's say. Similar thing is observed in the case of xylose and in the case of fructose. So let me say again what's going on here. All this data is the copy number of fructose transporters, of xylose transporters, of glycerol transporters for different growth rates, but there's always a single anomaly, although I wouldn't call it an anomaly. There's a single outlier. The outlier is the case in which the actual carbon source is the transporter, the carbon that's transported by the transporter of interest. So for example, when I have fructose around, then it overexpresses, if you like, the fructose transporter. At that growth rate, it says, okay, I'm going to bring on fructose because that is the carbon source that I have, and so it ramps up its production of the fructose transporter. Same for xylose and same for glycerol. So, so what are we supposed to conclude from this? And then we'll be done with this this vignette. What are we supposed to conclude? We're supposed to conclude that the bacterium certainly has enough transporters to bring on board carbon. And furthermore, we conclude that when it needs to add more transporters, there's certainly enough area in the membrane to add those transporters. How do I know that? Well, I know that for one thing because they're on the order of 100,000 membrane proteins per micron squared, and the area of the cell membrane, this bacterial cell membrane, is something like 5 or 6 microns squared. So that means that, and we're talking about of order of several thousand proteins out of a constellation of 100,000 per micron squared. So it's really a very modest burden on the membrane area. And so if the cell wants to add more transporters, there's sufficient room to do so. So, so that's the message. And so, you know, going back to our original hypotheses about bacterial growth and what's rate limiting, what I can say is that we can conclude that the first hypothesis, this one for sugar transporters, is probably not the rate limiting step. So in the next vignette, what we'll do is we'll explore ATP synthesis as a possible source of rate limiting.